Are you prepared? Yes,
I am.
please, Pieter from Amsterdam.
Thank you. Yeah, so it's a great pleasure to speak today. I have a few scientific heroes. One of them is Nikos. So I was really glad to get this invitation. Okay, so today I'm going to talk about how higher and lower areas interact with each other to make sense of the visual world. And this is a picture I'd like to show. So if you see this picture, I think it's visible, then you probably see a couple of animals. You don't have a hard time seeing that say, this is the nose of the animal that has this as one of its legs. So this is how we see, and what doesn't help is that vision actually starts with fragmentation. So neurons at early levels of the visual system at first sight seem only concerned about the information in their tiny receptive fields. So somehow the visual system has to build up these objects from these image fragments, and that is the process that I've been very interested in. Okay, and I'm going to claim today that this is solved by what we call an incremental labeling operation, where all these image elements that belong to a single perceptual object, the object of interest, are incrementally labeled with enhanced neural activity. Psychologists call it object-based attention. And actually, I would claim that this is the solution to the binding problem. So here I disagree with Samir. I think we actually solved it. Okay? So if this is the picture that comes into the system, then the visual system, if you're interested in the central zebra, will create a labeled representation where all these image elements, colors, shapes, motions, are labeled with enhanced activity. Okay? Now I have to convince you of this. So, first I'm going to talk about perceptual organization, where I'm going to try to understand the algorithms that the brain implements to make this happen. Then I'm going to talk briefly about visual awareness and also our goal to apply some of this understanding that we now have of the visual system to create the prosthesis for blind people. But I'll start with an introduction, and you have seen this, so I can go fast. So this is from a famous study more than 25 years ago by Velleman and Van Essen, showing all the areas of the monkey brain in different colors. This is from the same paper. So here you can arrange all these areas into a hierarchical scheme. We all know this. Now the idea is that when the new image enters into the visual system, there is this wave of feed-forward processing, where low-level areas extract simple features, such as the orientation of a bar. Neurons at intermediate levels, like V4, they would be tuned to feature constellations of intermediate complexity, and neurons at high levels would be tuned to image categories. Okay? Now this is then actually when the information reaches the highest levels, information processing need not be over, because there are also these feedback connections and lateral connections allowing for recirculation of activity. And in the second phase, when recurrent processing occurs, we believe actually that this labeling process will happen. And this is, I said it before, this is associated with what psychologists call object-based attention. Okay? Now let's see this at work then. So some of you may have seen this. So I now challenge you all. What I'm going to do is I'm going to present two curves. And one of these curves is going to make a connection between the red dot and either the green dot or the blue dot. And it's up to you to decide whether it's the green dot that is connected to the red dot or the blue dot. And if you think it's the green dot, you might want to raise your left hand. If you think it's the blue dot, you might want to raise your right hand. And you might want to be just a little bit faster than your neighbor. Okay? Are you ready? 1, 2, 3. Okay? Some people are working it out. Yes, it's not so difficult, right? So you just start here and then up. Okay, there you go. Okay? So you arrive at the green dot. Okay, now this is your time to take revenge. So now we start from this side. 1, 2, 3. Okay, 6, 7, 8, 9, 10. Okay, you see right hands? Yeah, this takes time. Okay, you start here, and then you go all along the curve. You go here, and then up, and there you end up at the blue dot. Okay, now this is a stationary image, right? All the features are there from the start, and this takes more than 10 seconds, which is an enormous amount of time for the visual system. So, of course, you're doing this with a strategy. The way you typically do this is you start fixating here, and then you make a sequence of eye movements. These will be saccadic eye movements. You cannot make smooth eye movements for a stationary image. You're going to make a sequence of saccadic eye movements along the curve that you're mentally tracing. Now, if all the time you're doing this, it's about 10% time is eye movement time, 90% time is mental work, mental curve tracing. So it's about 9 seconds. And what happens here is you probably start fixating here, and then you're mentally tracing a part of the curve because you make sure that your next eye movement will be on the same curve. So you don't go here, for example. So that implies that first you have to do some mental tracing before you make the eye movement. And so apparently these epochs of mental tracing, they add up to enormous amounts of time. So we wanted to understand this process, and this is a study that we did a couple of years ago, actually already many years ago. So we trained monkeys. They looked at a fixation point, then they saw 2 curves. One of them was connected to a red dot. This is the target curve. That connects the fixation point to the red dot. Here's another red dot that's not connected to the fixation point. This is a distractor. The monkey is still fixating, and he's mentally tracing the curve. Okay, and then after a delay, we remove the fixation point, and now he reports the red circle that had been connected to the fixation point with an eye movement. We record multi-unit activity using implanted electrodes. We started with single wires, now we use also Utah electrodes. And this shows you the recordings that we obtained. Actually, I was looking for synchrony. I was hoping to see that cells that respond to the same curve synchronize. Well, they do not. Synchrony is unrelated to binding. Okay? It's really something else. It's the enhancement of neural firing rates. And what we found is, so here the monkey fixates here at the fixation point. Here is a curve that then goes through the receptive field. His gaze is here, but he's mentally tracing through the receptive field. And here's tracing the other curve. And what we find is that the initial response is driven by the receptive field. It's the same. So that's what you see here. So this does not discriminate between target and distractor. It takes some time for this mental tracing process to reach the receptive field. And at that moment in time, the response elicited by a target curve is stronger than that elicited by a distractor. Okay, you see the same thing on the right. Okay, so by now we've not only recorded in V1, also in higher areas. And so in higher areas, of course, receptive fields are larger. So I'm going to talk about V4 and also frontal eye fields, an area of frontal cortex involved in the selection and generation of eye movements. So here receptive fields are larger, and frontal eye fields, they're actually quite large. So here the animal is planning an eye movement towards the frontal eye field receptive field. Here the animal is planning an eye movement elsewhere. So no surprise that there's more activity elicited by the target curve than by the distractor. But what's interesting is that there's this first epoch of firing where the frontal eye field neurons are driven by what's going on inside their receptive field, which is the same for the 2 stimuli. And it takes a bit of time, and then they start to select the end of the target curve for an eye movement. Now you see the same thing in area V4. First the visual response, then the selection process. You see the same thing in V1. You already saw this. First the visual response, then the selection process, right? So first you get feed-forward processing, and then you get this recurrent processing, interactions between different brain areas, jointly selecting what is relevant and what not. Okay, so first feed-forward processing, and then the recurrent processing. We also did some psychophysics in this task, and we find that, according to psychological definition, human subjects that trace a curve obviously also direct their attention to the successive segments of such a target curve, thereby, in some sense, integrating all these contour elements into a coherent representation of an elongated object solving the binding problem. Okay? So this is about curve tracing, and now I want to go to another task that we have studied quite extensively in the lab to understand the neural correlates of perceptual organization, which is texture segregation. And these are the results of Victor Lama, also almost 25 years ago. He demonstrated that neurons in V1, if this is their receptive field, the image element in the receptive field here and here are the same. But you see here they belong to a figure, and here they belong to a homogeneous region, could be a background. You see that the initial response in V1 is only informed about the image element in the receptive field, which is the same, therefore the response is the same. But it takes a little bit of time, maybe 90, 100 milliseconds, then the response becomes stronger if these image elements belong to a figure. Okay? So that's what we want to understand. So how does this work in the brain? And I'll talk about 2 processes that we think are important for this process, region filling and boundary detection. I'll say a few words about the role of attention. And then this has always been the holy grail in the lab, and I think we now have the data, and that is to demonstrate that these response modulations that happen relatively late in V1, that they're also causally involved in perception. And I'll also say a few words about the role of interneurons. Okay? So how does this work in the brain? Now this is a model. It's not so great on this slide, but I hope you can see that. So this is basically a neural network model where here are neurons tuned to the left orientation, maybe point it out like this. Here are neurons to the right orientation, so this is the stimulus. So they respond here to the figure, these guys respond to the background. Of course, in reality, these neurons would be intermingled in a single retinotopic map. Okay, here's a receptive field of a neuron that responds to the background, it's tuned to the right orientation. Now what we're going to implement to detect boundaries is iso-orientation inhibition. Neurons tuned to the same orientation are inhibiting each other. Okay? So this neuron now gets a lot of inhibition from its neighbor, its response is suppressed. Now, if a neuron has a receptive field that's on the boundary, the inhibition comes only from one side, so the response is less suppressed. Okay? And that then gives rise to the pattern of activity that you get a little bit extra activity at the boundaries because there's just a little bit less inhibition. Same happens for neurons tuned to the background, so they're more active in the surround and they have a little bit of extra activity at the boundaries. If you sum these two maps, then you basically get a frame, extra activity of the boundaries. Okay, that's how you can implement boundary detection. Now we're doing that at multiple spatial scales. V1 has small receptive fields. V2 has a little bit larger receptive fields, so you see that the frame is a bit thicker. And at some point you reach a level where the receptive fields have about the same size as the figure. So now you have a neuron with a large receptive field on the figure. All its neighbors tuned to the same orientation are silent, right? So this neuron is very active. It's called pop-out. Okay, so this is what you get by feed-forward processing combined with iso-orientation inhibition. You get boundary detection plus pop-out. Okay, and now we are going to do the region filling. So now in the feedback pathway, we are going to implement iso-orientation excitation. Okay, so here are neurons tuned to the left orientation, and they're going to help filling in this region, so that where this hole is in the frame, successively through feedback, enhancing the activity of neurons that are also responsive to the center of the figure, say in area V1. That's our model of how this works. Okay? What is nice about it is that psychologists suggest that this region filling, this boundary detection and pop-out process is pre-attentive. It doesn't depend on where you're interested in. Whereas this region filling process is our predictions to depend on attention. Okay? So that's what we set out to test a couple of years ago. And this was done in monkeys. They were looking at a fixation point. They saw a figure. I hope you can see it. This is a receptive field, not visible to the monkey. And there were two curves. Now, on some days, the animal was caring about the figures. He was supposed to make an eye movement to the center of the figure. These were the figure detection days. On other days, he could completely ignore the figures, and he only cared about the curves because he was doing curve tracing. Okay? This is work by Jasper Poort. So on the figure detection days, the animal makes an eye movement to the center of the figure. Curve tracing days, he simply ignores the figure and does curve tracing. And we varied the position of the figure. So if this is a V1 receptive field, the figure is, of course, larger. And here the receptive field is in the center of the figure. On another trial, there could be another stimulus where the receptive field was on the edge or in the background. And since we have the two orientations, we can make sure that what's going on inside the receptive field is always identical. And this is a replication of the Lamma effect. Basically, what we see is first a feed-forward response, and then later you get extra activity if the receptive field is on the figure, as opposed to when the receptive field is on the background. Now we're interested in these extra spikes. Where are these extra spikes? Okay, so we're going to subtract the background response from the figure response. That's shown here. And this is a bit of a complex slide. So what you see here is the spatial dimension. At 4, the receptive field is on the background. At 2, the receptive field is on the edge. At 0, it's in the center of the figure. Here it's on the other edge of the figure, and here it's on the background again. Remember that we varied the location of the figure relative to the receptive field across trials. This is the temporal dimension. So time 0 is when the stimulus appears. So what you see, this is V1. This is when the animal was doing figure detection. You see, it starts with boundary detection. You see an early detection of the boundaries, early response enhancement at the boundaries, and later filling in of the center of the figure with enhanced neural activity. Okay? So this is kind of consistent with the idea that boundary detection is feed-forward, and region filling depends on the loop through the higher visual areas. It has a longer latency. Now, this is when the animal was interested in the figure. If it was not, basically what we found is that this boundary detection process was still quite pronounced, but the region filling that depends on the loop through the higher visual areas is much less pronounced. Okay, we also recorded an area V4 where receptive fields are larger, and therefore we see early on also a response enhancement in the center of the figure. And we actually have now solid evidence that if you silence this, then also the response enhancement in V1 does no longer occur. Okay, so this demonstrates that this is really a genuine feedback effect. Okay, let me summarize this for you. So you present the Vega ground stimulus. Neurons start with their orientation selective responses. Early on, you get, probably through the iso-orientation inhibition scheme, you get a response enhancement at the boundaries, and then a later region filling in, which is pronounced if the animal is interested in the figure, but much less so if he's not, suggesting that non-attended figures are left in a somewhat more primordial state. Okay? So I think I covered this. And now, first, the big question. So we have always studied these subtle V1 modulations. Do they really matter for perception? Are they simply a correlate of processing in higher areas that feed back to V1, but it doesn't really matter for perception? Okay? So we wanted to do this with optogenetics, and we've spent some time in trying to get optogenetic inhibition to work in the lab. So far, we haven't really succeeded. We really wanted to silence V1 with optogenetics. So it didn't work in a monkey. So this is figure ground in a monkey. Actually, it's interesting to realize that this is not only in a monkey. You can get the same signal in different species. So we have a collaboration with neurosurgeons that see patients and treat patients for epilepsy, and they put electrodes in the brain, and we can sometimes record multi-unit activity through these electrodes. And this was a very seldom case where they put an electrode in area V3, and then we got the same effect, so more activity elicited by a figure as opposed to a background. Now here it's early on, because V4 receptor fields are relatively large, and the figure had about the same size as the receptive field. Okay, so but now we wanted to get the optogenetics results, so we went to mice. And mice also have figure ground. Okay, so in mice, activity elicited by figure is more than activity elicited by background. But now we wanted also to get mouse behavior, and that was very difficult. Okay, so you would do this in a monkey, he would get it on the first day. If you do it in a mouse, it takes forever. It took us actually 1 or 2 years to really understand what stimulus we should use, but in the end we got it to work. So here's the mouse, and he either sees a stimulus, a figure ground stimulus with a figure on the left, and he's supposed to lick a left lick spout. And if the figure is on the right, he's supposed to lick a left lick spout. And actually, this is the accuracy. So if there was no need for figure ground, just a contrast-defined stimulus, the animals are, well, what is it, maybe 90%. They're around 70% for the figure ground stimuli, and that is as much as you can squeeze out of a mouse. They just don't get better. But we have something to work with. Okay? So now the dream experiment. First, actually, we wanted to reproduce the figure ground effect by recording an area V1. So we use these laminar electrodes. This shows you just a V1 receptive field. You see it's about 20 degrees, which is about 20 times larger than in a monkey. And this shows you the activity elicited by these different stimuli. So a contrast stimulus, either something in the receptive field or nothing in the receptive field. The stimulus would be here, so you get nothing. Okay, here's orientation-defined figure ground. So you see you get an earlier response that does not discriminate, but relatively early on you get more activity elicited by a figure than by a background. Phase-defined, texture-defined. What is interesting then is you can also compute the latency of this figure ground modulation. So you subtract basically the background response from the figure response, as I did before. You see the visual responses after about 60 milliseconds. Then orientation-defined figure ground, 95 milliseconds. Phase-defined figure ground even a little bit later. Okay? So that all seems to work. Now we have the behavior at work, so now we're going to do optogenetic silencing. Do these late spikes in V1 really matter for perception? That's the question. Okay? So basically what we do, we use an optogenetic silencer, GT-ACR2, which actually knocks out all the spikes from V1 in all layers. So that's good. And you can knock out the entire response, okay, just silencing V1. But of course, we're interested in what happens then if you only let through the feed-forward response or successive periods of also this delayed response. Okay, so that's how we set it up. And this shows you the accuracy of I think, 5 mice. They're doing the contrast version of the task, so no figure ground required. If at time 0 we switch on the laser, we knock out the entire response. And the animals, actually their drop, this would be without optogenetic silencing, so they drop to about, 60% or so. So some of the accuracy in this task might actually be subserved by subcortical structures, because we are pretty good in knocking out V1 activity. But of course, we cannot completely rule out that we knocked out all the spikes. And you see that if you then postpone the silencing to about 62 milliseconds, the animals are half back to their maximal accuracy, and if you wait for, 70 milliseconds or so, then they could do the task. So just the feed-forward response seems to be sufficient for these mice to do a contrast detection. Okay? Now for figure ground, it's different. So this is orientation-defined figure ground. If we knock out the entire response, the animal are completely at chance level. And we have to postpone to about 100 milliseconds to get half maximum performance, and maybe 110 milliseconds before they can do it at ceiling performance. Okay, so we are really excited about this. Yes, the animals do need these late spikes to be able to carry out figure ground. And we have the other task, phase-defined figure ground, and you see then you have to postpone it even a little bit longer, maybe to about 160 milliseconds or so before they are at ceiling performance. Okay, that matches actually what we saw in the latencies of the figure ground modulation. So we're really excited about this. I think this is the first demonstration that late activity is necessary for figure ground modulation in V1. So these spikes are there for a reason. And of course, contrast detection can do without these delayed spikes. It just kind of, V1 is a relay, sends the information to higher areas, and they take care of the rest. Now, there's one thing that is so attractive with mice, and that is you have all these different Cre lines. So we wanted to see what the activity of the different cell types was in figure ground in mice. Okay, so and this shows you the theory about it. So you have the feed-forward response that arrives typically in the basal dendrites, feedback, layer 1, also some in layer 5. And there is this PV neuron that is thought to be important for gain control of the cortical column. Now, a nice paper that came out a couple of years ago by the lab of Scanziani suggested there is a particular type of interneuron, somatostatin-positive interneuron, that they suggested has a specific role in surround suppression. Okay, so the idea would be that they might be the ones to suppress this response elicited by a background. And another paper, a little bit later, by Jan Dan's lab, suggested, in addition, there is feedback to VIP neurons. Now, VIP neurons are a special brand of interneurons. Their specific task seems to be to inhibit the somatostatin cells. Okay? So the VIP neurons get active, the somatostatin cells decrease, and the cortical column is disinhibited. That's the idea. Okay, so it's really interesting to look at all these cell types. And they could actually, the VIP neurons could help with this disinhibition, getting extra activity at the figure location. Okay, so we started with the pyramidal cells. We are now doing 2-photon imaging, 2-photon calcium imaging. This is the figure response, this is the background response. So on average, there's more activity for the figure. It looks like a weak effect, but we're actually plotting it on a logarithmic scale. So this is a substantial effect. And if you average across all the V1 neurons, so more than 1,000, the standard deviation is plotted here. It's hardly visible. You see that the figure indeed elicits a much stronger response than the background, as you would expect. Okay, so that's what the pyramidal cells do, not unexpected. Okay, now we're going to look at the other interneuron types, so somatostatin neurons, and this is a nice result. They do the opposite. Somatostatin cells have more activity at the background than at the figure location. VIP cells, they're supposed to supply the disinhibition. They have more activity for the figure than for the background. And PV cells, they actually follow the pyramidal cells. They have a very strong figure ground modulation and much more activity for a figure than for a background. This is predicted by some of the models on this. Okay, and this shows you the statistics. So decent figure ground modulation for pyramidal cells and VIP neurons, opposite for somatostatin cells, and very strong positive figure ground modulation for PV cells. And so basically what we find is that these image regions that are labeled with enhanced activity, they're associated with extra activity of pyramidal cells, VIP cells, and PV cells. Those image regions that are suppressed, they are associated with extra activity of the somatostatin cells. Okay. Okay, now I would like to say a few words. It's a similar type of reasoning about feed-forward and feedback connections, but we recently carried out a study where we unexpectedly found some effects that are related to when a stimulus does or does not reach conscious awareness, or at least conscious report. So I'm going to try this out with you, but it's kind of unpredictable with this projector. So I'm going to show to you a stimulus. If you see it, please raise your hand. If you don't raise your hand. Okay, so 1, 2, 3. Okay, yeah, that was easy. Okay, now I'm going to make it more difficult. Okay, are you ready? 1, 2, 3. Who sees it? Some doubts. Once more. 1, 2, 3. Yeah, that was easy. Once more. 1, 2, 3. Some people saw it. Actually, there was a stimulus in the first and the third presentation. In the second and the fourth, there was no stimulus. So those people who raised their hand, they had a false alarm. So thanks for doing that, because I needed it for my explanation. Okay, so this is signal detection theory, a very famous theory, accounts for quite a lot of data. And the idea is that there is somewhere in the brain an internal representation of signal strength that has perhaps a Gaussian distribution, could be another distribution. Okay, if there is a stimulus, then you get this distribution. If there's no stimulus, you get a distribution with a lower mean, according to signal detection theory. So across trials, you're sampling from these distributions. If there's a stimulus, maybe the internal representation of signal strength is here. And then, if the task is to report whether you saw something or not, in the signal detection theory, there's a threshold. And if activity is above this threshold, you would say yes. And if it's below the threshold, which may also happen sometimes because it's stochastic, you probably say no, and that will be counted as a miss. If you say yes, it will be counted as a hit. Now, on trials without the stimulus, you're sampling from this distribution. So if you say no, that would be a correct rejection. And if you sometimes, by the noise in your system, would say yes, even though there was no stimulus, that would be a false alarm. Okay, so this is when the stimuli on average are quite close to invisible. Now, if the stimuli are more clearly visible, the signal present distribution shifts to the right. And now if you apply the same threshold, you'll see you still get quite a number of false alarms. So you might want to raise your threshold a bit, because you're not going to have a lot of extra misses. Okay, so maybe this is a better location of the threshold. If you apply this threshold here, of course, you're going to have quite a lot of misses. So this is what signal detection theory is good at. It's kind of predicting how human observers set their threshold as a function of various variables, one of them being the average strength of the stimulus. Okay, now the questions we asked here is, where is this signal, this internal representation of signal strength? And what causes this variability, the width of these distributions? And this is related to the global neural workspace theory, where the idea is that if you present a weak stimulus, it gets propagated to higher areas. And then, according to the global workspace theory, there is, so by Stander Hane and his collaborators, there is this magical process that they call ignition. If that happens, they think it relies on the recurrent interactions between lower and higher brain areas. And then even if you take the stimulus away, the signal is still stabilized, right? So it's still reportable. That's the idea. Okay, so if the stimulus is not propagated well enough to those areas that cause ignition, then it will not be reported. It will result in a miss. And if it does give rise to ignition, then it will be a hit. Okay, so we trained monkeys on the same task you were doing. So in 50% of the trials there was no stimulus. Now to have the monkeys report that they didn't see anything, we always had this dot on the screen that we call a reject dot. So if the animal wants to report there was nothing, he just makes an eye movement there. And if there was a stimulus and if he wants to report it, he makes an eye movement to the location of the stimulus. Okay, very simple task. And we have the animals on the staircase, so you vary contrast, and you get, of course, a very well-behaved contrast response function in behavior. Okay, we have two thresholds here. We have one threshold where the animal reaches 80% correct. If the stimulus is easier than that, we call it an easy stimulus. The second threshold is at 40%. So if the animal is below that, we call it a difficult stimulus, and in between we call it a stimulus of intermediate difficulty. And then we'll record from area V1, V4, and frontal eye fields. And this is in V1. This is for the three categories of stimulus difficulty. And basically, and it was somewhat surprising, is that what we're doing here is we're sorting trials, but we're sorting trials with precisely the same contrast. So the distribution of contrast is matched between hits and misses. Okay? Now, apparently, on those trials where the animal fails to see the stimulus, not as much activity is elicited in V1, suggesting that some of this variability is already happening downstream from area V1. Very surprising. You see, at the intermediate level, the difference, at least proportionally, is less, and at the easy level, in proportion, it's even less. So suggesting that, what is surprising is that easy stimuli that are missed, they actually in V1 elicit more activity than difficult stimuli that are seen, suggesting that it's not V1 that determines what the threshold for conscious perception is. Okay? We get the same result. Oh, we actually can then express this as the miss fraction. That is the fraction of activity that remains on the missed trials. Okay, so you see that the miss fraction, basically the proportional size of the red curve, increases with stimulus contrast. We then do V4. We basically get the same result, maybe a little bit less activity on difficult stimuli with a miss, as if more activity got lost at the level of V4. But again, in V4 you see that easy stimuli that are missed still elicit more activity than difficult stimuli that are seen. Again, implying that it's not V4 that is determining whether a stimulus will be consciously perceived or not. Okay? And then we record from the dorsolateral prefrontal cortex. So you see that the miss fraction in V4 increases with when it becomes less difficult. And so here in dorsolateral prefrontal cortex, we are actually in an area that generates eye movement. So this is frontal eye fields. So we're also recording from neurons that are encoding the plan to make an eye movement. So we're not dissociating here the conscious percept from the plan to make an eye movement, just to make that clear. Okay? Now you see in dorsolateral prefrontal cortex, it's really all or nothing. So irrespective of the difficulty of the stimulus, if the monkey is going to report it, you get a lot of activity. If he doesn't, you get less activity. Although you see that if in the easy stimulus that are missed, there is a little bit of activity elicited in the frontal eye fields, but apparently not enough to cause this ignition process. So here you see that the miss fraction is uniformly small. So here you can put a threshold. If activity crosses this threshold, then it might lead to ignition, and the stimulus will be reported. And if it stays below the threshold, it will not be reported. And we also, of course, record during those not so many trials where the animal commits a false alarm, and we compare them to correct rejections. So the correct rejections here are in black, and the false alarm are in blue. Okay, so what we seem to be seeing here is sort of spontaneous ignitions. Now, if there's no stimulus, there's not a natural time zero. So these spontaneous ignitions can then happen, of course, at different times. And we think that the activity then goes up. And if you then average across trials where these maybe relatively sudden increases, but happening at different times, then in the average that will give rise to a ramp. That's what you see. Okay? And this is quite consistent then with one of the places where you can find this internal representation of signal strength. I don't claim that this is the unique location, but this is one of these regions where you will find a signal where activity is strongest on hits and false alarms, and weakest on misses and corrected rejections. Okay, so what we seem to find then is that activity seems to get lost. Weak stimuli get lost at low levels of the system. Easier to see stimuli, high contrast stimuli seem to be activating V4, but seem to get lost at a later stage in the processing chain. But of course, not the only way to get to the frontal eye fields or to the frontal cortex is using cortical-cortical connections. There are also subcortical routes, right? So we next asked, is variability in cortical-cortical propagation of activity, is that in part responsible for this variability in the report? And we therefore did a variation of the same experiment, but now we use microstimulation. We directly activated neurons in area V1 with microstimulation, and we recorded activity in area V4 with these Utah electrodes. Okay, so the task is almost the same. So this is the receptive field. It's not visible to the monkey. On 50% of trials, we don't give microstimulation to primary visual cortex, so the animal makes an eye move to the reject dot. And on 50% of trials, we give then microstimulation, and we now have the current strength on the threshold, on the staircase. And so we get decent-looking psychophysical performance. And then this shows you the configuration of receptive fields. So this is the V1 receptive field we're stimulating, and this is the V4 receptive field we're recording from. And these are individual trials, recordings in V4, and if we give a clearly subthreshold stimulus in V1, then typically we see a V4 response that looks quite like a visually driven response on basically every trial. And we then basically get the same effect when we compare stimuli that are microstimuli that are perceived by the monkey. They elicit more activity in V4 than those that are not perceived. And you also see this increase in miss fraction. So here, stimuli that are missed by the animal, but that are should be easy to see, they elicit actually more activity than the difficult stimuli that are perceived, suggesting that also in this task, information can also get lost later. Okay? Now, I showed these data to Stan de Hane, and he was really excited, and he said, oh, we can model this. So this is a bit embarrassing, because he made a model with 5 stochastic neurons, only 5 neurons. That's the whole brain, okay? And the model works as follows. So activity comes in the LGN, then goes to a single neuron in V1, to V4, parietal cortex, frontal cortex. Now, reciprocal connections between those regions are a bit stronger than down here, and so strong that in some cases, they can give rise to self-sustained activity, ignition. Okay, if you then take away the stimulus, the activity is maintained, and that will be a scene trial, a hit. On other trials, because these neurons are stochastic, it stays below the threshold here to cause this self-sustained activity. If you take away the stimulus, the activity goes down. Okay, that would be a miss. And this shows you the activity profile. I'm going to go a bit fast now. But basically, we were replicating all the things that we saw during meticulous recording over many years. He probably did this in a Sunday afternoon. Okay, so we also get the same effect on false alarm trials and correct rejection. So this is the model, this is the data, and I was really embarrassed. It's a really nice fit. Okay, so I want to spend the last 2 minutes or so talking about the visual prosthesis, so I go a bit faster now. So we see that weaker stimuli get lost at lower hierarchical levels than stronger stimuli. False alarms can be explained by spontaneous ignitions. And, well, basically the global workspace theory gave quite a nice account for the pattern of results in this task. Okay? Now the last 5 minutes or so, I'm going to say a few words about applying these... I have a very severe-looking chairman, so I'll go very fast then. Is that okay? No, it's not okay. Okay. So I go very, very fast now. I just want to show you this. So we are now implanting in the monkey 16 Utah probes. Okay, these are 2 monkeys. Okay, in V1, 2 of them in V4, and this shows the receptive fields. This is monkey 1, this is monkey 2. And if we then move a light bar across the screen. This is now plotted in the visual coordinates. You see that the light bar is trailed by a wave of activity, right? So this is Hubel and Wiesel times 1,000, because we have 1,000 electrodes. We then train monkeys on the same task that I showed you before, but we now can, of course, stimulate 1,000. We have a lot of choice. So this is the eye movements of the monkey, and this is the electrode we're stimulating. And we're basically able to produce these elementary percepts for hundreds of electrodes, which I think is quite promising. And then we train the monkeys on a task in which they need to recognize one of 16 letters of the alphabet. Okay, so here the monkey sees a letter D, and then he reports it by making an eye movement to the corresponding letter. And what we then do is we don't stimulate a single electrode producing a single phosphene, we produce, say, 10 or 15 electrodes, and we know that their receptive fields are on the shape of the letter A, so basically the black receptive fields here, hoping that the animal will make an eye movement to the corresponding letter. And this shows you the results, and then I will be done. So here we're stimulating a bunch of neurons in the shape of letter O. Hope you can see it. It's a bit dim. So basically demonstrates that we can produce quite fine-grained percepts by stimulating patterns of electrodes in primary visual cortex. And this basically shows that across sessions, the animals are clearly above chance level, actually from the first trial onward. Okay, so that's the plan, to build a system where you have an image processing unit, this is a camera, then you send those signals to the brain from microstimulation, and that's something that we are trying to get to work in the next couple of years. So I would like to end by showing the people who did the work. I'm not going to list them in the face of time, but it's really a big team effort, and I think I mentioned many of the people who were involved in the individual projects as I gave my presentation. So thank you very much.
Thank you very much. There are, of course, many questions. Please be short, precise. Okay.
So, Pieter, I'm asking you a question by hearsay, which is a little unreasonable, but I recently heard Doris Tsao talk about some behavioral results she's obtained with teaching mice a similar visual discrimination to yours, a figure ground discrimination. And what she found was that the mice never actually learned the concept of figure ground. They learned each discrimination task separately. They had no savings across the tasks. And that leads me to wonder, which is what the real question is, do you have reason to think that the mice really have a notion of a figure, that they have object vision in the same sense as primates? Because they may have mechanisms that look the same, but that doesn't mean they actually use it to see objects with. And I wonder what your thoughts are on that.
So I think our data are actually more encouraging than Doris's data. I don't know about these data, but I showed you that the mice did first orientation defined, then face defined. They generalized fairly quickly. When we presented the textures, I think they got it on the first day.
In your early experiments on the monkeys where they're doing the curve tracing, they're required to continue fixating during the trial. But have you looked at the microsaccades to see if they provide any indication of what you're seeing in the visual response as they get to a certain part of the curve?
Yes, so there are two things about that. The first thing is that as a control, because small variations around the fixation point could already cause difference in firing rates. So we did extensive controls for that. It cannot explain the result we're seeing. And I mean, also you would not expect a consistent effect across all the neurons. Some neurons should go up, some neurons should go down. So I think we can completely exclude that confound. The other thing is, and that is actually related to something that Tony said, what happens across eye movements. So we did a study in which the monkey was looking at a fixation point, then there was a curve, and then he was supposed to make an eye movement here, and then the curve went on in this direction as a target, and below there was a distractor, or the other way around. And then the target went down, and the distractor went straight on. And then we had many animals were supposed to make an eye movement to this dot, and then to the second point on the target curve, so either there or there. What we found is that when you have a V1 receptive field that is not yet on the curve, that is brought to the target curve by the first saccade, it has the response modulation earlier than if you present the stimulus for the first time on the screen. That's interesting, right? As if there are savings across eye movements that kind of quickly after the eye movement compensate for the fact that an eye movement happened and tell the right V1 neurons, hey guys, now you're on the target curve. We also did a version in which during the eye movement we flipped connections. What you then see is that the early response of the V1 neurons that lands on the curve, that before the change was a target curve, they first have an enhanced response, as if they have a memory of that this is a target curve, and then it takes some time and then they flip.
Yeah, thank you very much for this really comprehensive and impressive talk. I have just a quick question regarding the prosthesis stimulation. In which area, in which layer do you stimulate, and do you think that the layer in which you stimulate is important or not?
Yeah, that's a good question. So we are using these Utah electrodes. They have a length, in this particular case, of 1.5 millimeters. So I think that would typically be lower layer 4 or upper layer 5. But of course, we have not perfect control in how they are inserted. So some of them might be a little bit shallower because they're not inserted perfectly. Now there has been quite some good work of Edward T. Hofnick, who looked at the influence of microstimulation on primary visual cortex in the different layers. It seems that you can elicit these percepts in all the layers if you want, but there seems to be the thresholds seem to be lowest in layer 5. But there's basically a profile, and other than that, there seems not to be all that many differences between the results that you get when you stimulate these different layers. Only a difference in thresholds.
Just a very quick question. I find it very interesting that for the optogenetic experiments, that it's a dissociative vision mechanism that, from my understanding, allows the gating of the center and surround. And I was wondering if you have done this coupled to the electrophysiology and thereby vary the parameters of the stimulation and test the network and how it can bias or shift the likelihood of detecting these stimuli. Because I figured this is quite. I mean, given the genetic tools that Cree driver rely on, this would be a very nice paradigm for testing these hypotheses.
Yes, no, we haven't done that. That's something we're thinking about. Scanziani also is carrying out experiments in this direction, looking at the interneurons in figure ground. And they have had several posters on it at the Society for Neuroscience meeting. And what they seem to find seems to fit the story, so that it's the VIP neurons that indeed inhibit the somatostatin cells. So, of course, the difficulty with those experiments is you should probably not bring the system out of its dynamic range. So if you give a very strong optogenetic stimulus, completely silencing the VIP neurons, for example, the cortical column might be out of its normal operating mode. So that's always a bit difficult there. And you want to see effects, so there's also always a trade-off. With very strong stimulation to see effects and weak stimulation to really see the cortical column in its normal operational mode. Now, the other question implicit in what you asked is, looking at behavior, I mean, that's a real cool question. Jan Dan has also done experiments like that, maybe. I think she will be talking about sleep at this conference, but there she also has some really remarkable effects. So I think if you activate the VIP neurons, things like working memory become even better in mice.
The stimulation results in a monkey, the last experiment you talked about, in theory it can be explained by paired associative learning. You stimulate something and you associate it with another stimulus. So a critical experiment is generalization, basically. Is that the case?
Yes. Very good question. So I didn't have time to tell the full story, but so we start the animals on letters like I showed. But then we break them up and we try to make them look like we think that these phosphine patterns will look like. So we also train the monkeys on A's that on the screen look like a number of dots that are on the contours of the A. And now this is a very long duration process over many, many months. Now your question is, do they learn these arbitrary patterns and then quickly learn to associate them with the required response. So to address that point, we also took arbitrary letters that the monkey had not learned about as a control. We did that visually, and we did that also with microstimulation. And the animals are completely at chance level. Okay, so these, 200, 300 trials that we use to test the monkeys are just not enough time for the animals to associate these somewhat arbitrary patterns then, in that case, to an eye movement response. That just takes more time. So I think we can rule that out.
Thank you very much. Thank you, all the speakers of this morning. It was, fascinating. The last presentation showed quite clearly that the laser pointer is one of the most useless inventions of modern time, because you never know where he's going to direct in the next step, showing here or there. I think what we need is a laser pointer like Obi-Wan Kenobi used in Star Wars, where you actually can see, like a classical stick, so that the eye can actually follow where you're going to show. So we participate in a very interesting experiment on unpredictability. Thank you very much, and we have a lunch break now.